Hello everyone, let us look now at nucleic acid structure and function. The nucleic acid, we saw they have the common pattern like the carbohydrates and proteins, where they have a monomer that comes together to give us a polymer of either DNA or RNA. The interesting thing about nucleic acid, their monomer is not one molecule, it's made up of three components. We have a sugar, which is a 5-carbon sugar as we had mentioned, which we call ribose. It can be either ribose or deoxyribose depending on whether the nucleic acid is DNA or RNA. In addition to the sugar, we have a phosphate group and a purine or a pyrimidine base. Okay, now if you notice the sugar ribose, its carbons are labeled starting with carbon number 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Why they are labeled with prime is to differentiate them from the molecules that we find in the purine or the pyrimidine bases. The difference between ribose and deoxyribose comes on carbon number 2, where the OH group, which is here shown is pink, can lose its hydrogen group to be deoxyribose, as we see in DNA, or it can stay as ribose as we see in this structure. So, for it to remember here, the monomer of nucleic acid is the nucleotide, which is made up of a nitrogenous base, a pentyl sugar, and a phosphate residue. The nitrogenous base we have mentioned they can be of two types, purines and pyrimidines. If we can start looking at the pyrimidines, there are three types also, the cytosine, thymine, and uracil. Cytosine is found in both DNA and RNA. However, thymine is only found in DNA, while uracil is only found in RNA. We don't have uracil in DNA. The purine bases are of two main types, and they are found in both DNA and RNA, adenine and guanine. Two main types, adenine and guanine. Okay, now a breakdown of adenine and guanine will ultimately give us uric acid, which is passed out in urine. But in the process of breakdown, there are intermediates which include hypoxanthine and xanthine. And these ones are good indicators. We're going to meet them again when we're going to talk about gout, where there's increased production of uric acid. Then we have unusual bases. These unusual bases, we have modification of the bases, usually for the purposes of identification. The most common form of modification of the bases is through methylation. For example, you can have different forms of methyl adenine, methyl guanine, or dimethyl adenine, which can be found in messenger RNA molecules. And this is to allow the messenger RNA to exist for a period of time so that it can be transcribed at the ribosomes. We have methylcytosine, which is found in both bacterial and human DNA. This form of methylation helps in the identification of our own DNA versus foreign DNA. We have other bases that we found in plants. I mentioned them here because they have pharmacological properties. For example, tea, the one we take, has a base referred to as theophylline, commonly known as theophylline, but it's actually a modified xanthine into the dimethyl xanthine. Cocoa has a base which is referred to as theobromine, but it's 3,7-dimethyl xanthine, while coffee contains 7-methyl xanthine. We know these modified bases that are found in plant products have pharmacologic properties in humans. Then here is an interesting slide which shows us nucleosides and nucleotides. Nucleotides are the ones we have said are the monomers of nucleic acids, and we have mentioned they have a purine, a base, a ribose base, and a phosphate. But for nucleosides, they lack the phosphate group, meaning they are only made of the purine or the pyrimidine base and the sugar. Okay, so that is a big difference, and this difference is what contributes to the differences in the names here. For example, if you see adenosine, we know this is a nucleoside. It is made up of ribose and adenine base. But if you see adenylate, this is a nucleotide. It is made up of ribose, phosphate, and adenine. Okay. Then we have internucleotide bonds. We had mentioned earlier the bonds that hold the bases together are the phosphodiester bonds. If we look at the first diagram, the DNA, we can see the nucleotide that has adenine is linked to the nucleotide that has thymine through a phosphodiester bond. And the phosphodiester bond are linking the phosphate bond that is present on carbon number 5 on the nucleotide that has thymine to the hydroxyl group on carbon number 3 of the nucleotide that has adenine. So this is how the phosphates link with the hydroxyl group in the sugar. So we refer to this backbone as the phosphoribose backbone. And the linkage, the bonds are phosphodiester bond. But because of this, we can see at the terminal, we have a terminal at the 5' end of this nucleotide, we have a free phosphate group. So this is very common. We know our nucleotide chains have direction. The 5' end has a phosphate group, while the 3' end has a hydroxyl group. Similar things also happens in RNA. So please remember the phosphodiester linkage is linking the phosphate group of the bottom nucleotide to the hydroxyl group of the top nucleotide. And for we have a sugar, a ribose phosphate backbone. We have some nucleotides that serve as synthetic derivatives. For example, anti-cancer agents such as 6-thioguanine and 6-mercaptopurine and azathioprine are all anti-cancer agents. We know the drug allopurinol that is used in the management of gout that inhibits uric acid formation is actually a drug that has similar structure to xanthine and hypoxanthine, and it blocks the enzyme xanthine oxidase that is used to make uric acid. We have antiviral agents such as cytarabine and vidarabine, in which instead of ribose as the sugar, we have arabinose being used as the sugar. And then we have aminophylline and theophylline. These are used to interfere with the catabolism of cyclic AMP, and we know they're very important in the management of asthma. So the AMP is very important in the regulation of metabolic pathways. Okay, so it plays a very important role in the regulation of metabolic pathways. We are going to see when the energy level is low, metabolic pathways that activate energy. When the AMP concentration is high, metabolic pathways that stimulate production of energy are activated. When the energy level is low, or the AMP concentration is low, these pathways would be inhibited because AMP, ADP, and ATP exist in a ratio. So if AMP is high, it means that the ATP is low. Okay. So as I was telling you about cyclic AMP is a second messenger which is derived from ATP, and it is very important. We do realize that the cell membrane is a phospholipid bilayer, and some of the molecules are not able to enter inside the cell. So things like some hormones, they bind to receptors on the cell membrane, and they cause an intracellular cascade that causes the effects their changes. And one of those methods that can be used is conversion of ATP to cyclic AMP by the enzyme cyclase CAMP cyclase. So the CAMP will now activate changes into the cell that will cause an effect of the enzymes. For example, things like adrenaline and glucagon, hormones like adrenaline and glucagon, they bind to their receptors on the cell membrane, and they activate the production of cyclic AMP. The other nucleotide of biological importance is GTP. GTP is involved in energy transfer within the cells, and they serve as a source of energy for protein synthesis and even in other processes like gluconeogenesis. A new molecule of glucose is formed. It is also very important in translation. We're going to see it in molecular biology, and it serves here as a source of energy instead of ATP. It also used as a substrate for energy for the synthesis of DNA and RNA, and it also serves as a source of energy for polymerization of microtubules. So it also has very important roles. Interestingly, GTP also behaves almost the same as ATP, but ATP is more widely used. So now let us look at the structure of nucleic acid. Similar to proteins, the nucleic acids are organized in their primary structure, where we have the covalent structure made by the nucleotide sequences, how they are organized. And we see the bases we can now organize the bases like A, T, G, C. Then we have the secondary structure, which is formed by the folding or the interaction of the nucleotides. And then we have the tertiary structure, we have the complex folding of the nucleic acid chain into chromosomes in the eukaryotes or in the bacteria. So unlike protein, it only has 3 levels of organization: the primary, the secondary, and the tertiary. So now we can look at the DNA structure as the first nucleic acid we are going to talk about. So the DNA structure was postulated by these two gentlemen, Watson and Crick. They came up with a hypothesis of how the DNA structure appeared. So the hypothesis had to be in such a way that the DNA molecule was able to open up to allow that information that they are stored to be read, and then it can close up so that this information can be available. So Watson and Crick, they came with this model in 1943, which they called it the 3-dimensional model of the DNA structure. The first thing they said is that the DNA consists of 2 helical chains that are wound around the same axis to form a right-handed helix, as you can see in this diagram here. Okay, we have 2 helical chains. Okay. The other thing they mention is that the outside of the helical chain consisted of the deoxyribose and the phosphate group, as you can see in the diagram, while the bases, either the purine or the pyrimidine bases, were on the inside, as you can see here. Okay, so because the bases are hydrophobic, they don't like water, they're going to hide away from the water. Okay, so we have the phosphates and the ribose sugars on the outside and the bases in the inside. The other thing they mention is that the purine and the pyrimidine bases of both strands are stacked inside the double helix in a planar way. So we have the 2 chains, and then here inside we have the bases that are stacked inside the double helix with the hydrophobic near planar ring structures very close together in perpendicular. This diagram is showing it very well. You can see the bases in the center looking as if they are on top of each other. Now, the helical structure has 2 grooves. There is one big groove which is called the major groove, and there's a small groove on the end which we refer to as the minor groove. Okay, there is a major groove and a minor grooves. The grooves are very important because this is the point of the interaction between the DNA and proteins that we are going to see. This facilitates the folding. Okay, then the bases in the inside, they tend to base pair through hydrogen bonds with other bases, and there is a pattern. Thymine always binds with adenine, T and A, and they have 2 hydrogen bonds, while guanine always binds with cytosine, G and C, and here we have 3 hydrogen bonds. So please remember, so there is intra-binding hydrogen bonding of the bases that are within the double helical structures, okay? And hydrogen bonds, as we had mentioned earlier, they are not very strong bonds. They are formed between the interaction of attraction of electrons, and so they are not very strong bonds. So they are strong enough to hold the molecule together, but they are weak enough to be broken up and the information to be read. Interestingly also, the strands of DNA, they run in antiparallel direction, meaning one strand runs from the 5' to 3' direction, while the other strand runs from the 3' to the 5' direction. So they are all looking in different directions. That's what they mean in antiparallel. So this was a really good model for DNA because it could allow the DNA molecule to open and we read this information, which is stored in the form of the orientation of the base pairs TGCA. That is information that can be read and interpreted into a protein. And then after it is read, it is closed again. Okay, so this model of Watson and Crick is very widely accepted. Now, there are some rules that help us to understand the structure of DNA. They are referred to as Chargaff's rule. These rules were created by Chargaff as people were trying to understand the structure of DNA. And this was based on his observations in the environment. One of the rules is that the base composition of DNA generally varies from one species to another. Look at this word, base composition. When we talk about the base composition, we talk about how many adenines are there, how many guanines are there, how many thymines are there, how many cytosines are there. So this will vary from one species to another. Okay. Another interesting thing is that the DNA specimens isolated from different tissues of the same species have the same base composition, and this is very interesting. So our cells have the same DNA. So even if you take a cell from the ear, a cell from the mouth, a cell from the liver, if you isolate the DNA in that cells, so long as they are from the same species, they will be the same person, they'll have the same base composition. So it's not possible you get like DNA from the toe and DNA from the tongue are different. No, they are the same. Okay, so this is very important because I had some students saying that the DNA from the liver is different. No, the DNA is the same. The only difference is which part of that DNA is read. So I want you to think about as DNA as a library. In the library, there are many books. Okay, now the parts of the liver go and read liver. The parts of the tongue go and read tongue. But you have the whole library. So this library is copied. The whole library is copied and is there present in all the cells. And each cell goes and reads their part of the library. That's why it looks like that. That is specialized to be either be a liver cell or a bone cell because it's reading that section of the library. So the DNA from different tissues is exactly the same. Then the base composition of DNA in a given species does not change with an organism's age, nutritional state, or changing environment. Now this is very important. I had mentioned earlier your DNA does not change. Whether you move from Nairobi to America, your DNA will stay the same. If you're 15, if you're 30, 45, 90, the DNA composition does not change. Nutritional state, if you eat a lot of food, the DNA you are born with will stay the same. Do you have mutations? Yes, but these do not occur in the living organisms. They do occur in the incoming children. And then the fourth rule of Chargaff's is that in cellular DNAs, regardless of the species, the number of adenosine residues is equal to the number of thymidine residues. We saw that because A and T always base pair. So if there are 10 adenines, there are 10 thymines. Guanosine and cytosine also base pair. If there are 10 guanosines, there will be 10 cytosines. So because of this, it gives us a sum of A plus G is equals to T plus C. So how is DNA organized? So we know that the DNA in our cells is organized in chromosomes, and that's when we talk about humans. We have 46 chromosomes or 23 pairs. So our DNA, the double helix, you can see it folds around proteins, which we refer to as histones, to create a structure which is referred to as a nucleosomes. Then the histones further fold on themselves through several levels until they become the chromosomes. The usual forms we can see in the cell during mitosis is a very highly folded form of the DNA, which can be seen even through the microscope. Okay, these ones can be seen during mitosis. But when the cell is not preparing to divide, the DNA is slightly opened, okay, and we can see like spots or a dark spot in the nucleus. So in prokaryotes, the genome is usually a singular circular chromosomes, which is different from eukaryotes, which you can have several chromosomes, okay, and usually they're in a set of they're a set, okay. Mitochondria and chloroplast sometimes have DNA, but their DNA is usually circular, okay. So we have said the DNA is wrapped around histones, and then these histones are the ones that compact due to different levels until they give us, through coiling and supercoiling, to give us the chromosome, which can be seen at the mitosis. Further on, each chromosome structure, if we look at it, which the two chromosomes, they have a center part where they meet, which is referred to as a centromere. And each chromosome is referred to as a chromatid. Okay, so a chromatid is one of the attached chromosomes. And they have a central constricted area, which we refer to as the centromere, where they join. Further, if we think about the chromosomes we have in the human body, we can think about them in two types: the autosomes and the sex chromosomes. The autosomes, okay, there are other chromosomes that are not the sex chromosomes. The sex chromosomes are the ones that determine the sex of the organism, and they can either be male or female, which is X and Y, okay. So a female has XX and a male has XY. Finally, we have the ribonucleic acids. The ribonucleic acids, RNA refers to R ribonucleic acid, and the bases that we find here are adenine, uracil, cytosine, and guanine. Here we don't have thymine. Unlike DNA, ribonucleic acids are usually single-stranded, and they contain the sugar ribose. What the ribonucleic acids do, they carry information from the nucleus to the cytoplasm, which is the site of protein synthesis. And there are three main RNAs: mRNA, tRNA, and rRNA. M is messenger RNA, T is transfer RNA, and R is ribosomal RNA. Okay? Now, the information that is contained in DNA in the form of AGCT is able to be translated from AGCT to the protein language by the ribosomal RNA. Okay? And so the processes of transcription and translation mainly involved the RNA. So what are the functions of RNA? RNA has many functions, but as I mentioned, RNAs are mostly involved in protein synthesis, where they're able to control the assembly of amino acids into the protein. Okay, and each type of RNA is specialized in a different aspect of protein synthesis. So in this slide, we can look at the differences between DNA and RNA. You look at them slowly in terms of where DNA is found, where RNA is found, the structure. DNA is double helical, RNA is single-stranded. The sugar molecule in DNA is deoxyribose, the sugar molecule in RNA is ribose. Okay, the base composition, DNA is AGCT, but the base composition is AGCU in RNA. Okay, so we know the Shagap rule of A plus G is equals to C and T only applies in DNA. In RNA it does not apply because, as we said, the RNA is single-stranded, and sometimes it folds itself, but it's always single-stranded. It folds upon itself, so it might appear to have sections of double-stranded, but it's not because there are two chains, it's because it has folded along itself. Okay, so let's look at the messenger RNA. The messenger RNA is the one that is able to carry that information that is stored in DNA and takes it to the nucleus. So what happens during the process of transcription, the DNA chains open up and the messenger RNA is synthesized along, containing a corresponding sequence to the DNA. So if here there was A, messenger RNA will have T here. So this information is the same as the information that, not exactly the same, but it's complementary to the information that is there on the DNA. Once the messenger RNA is synthesized, it is packed and translated to it is transported to the cytoplasm, where now it can be used for making protein synthesis. So the information in nucleotide is usually coded for every 3 nucleotides. For example, AGC. So every 3 nucleotides are referred to as a codon, and a codon codes for 1 amino acid. For example, phenylalanine is coded by UUU. We're going to learn it as the genetic code. Okay, so we have slight differences in messenger RNA of eukaryotes. The messenger RNA is synthesized as a pre-RNA, and it undergoes some post-transcriptional modifications to become a mature RNA, which can be carried to the cytoplasm for the protein synthesis. One of the things is that the eukaryotes, the messenger RNA, both has coding regions and uncoding regions. The coding regions are called exons, and the uncoding regions are called introns. So once this messenger RNA is made in eukaryotes, it has to undergo some modification to remove the non-coding regions referred to as the introns for them to be removed. Then we have the transfer RNA. So the transfer RNA is a very interesting molecule. If you look at it, it looks like a clover leaf, but it has 2 ends. It's actually a very small molecule. It only has 80 nucleotides. It has 2 ends. On one end, it is able to link to a particular amino acid through a ester linkage. On the other end, we have the anticodon arm, which is able to recognize the codon on messenger RNA. So actually this one is the one that translates. On this one end, it can see the codon and read, like in this case, GCG, and it can be paired with it. And on this other end, it has that particular amino acid. And so it brings the amino acids close together to form the peptide bond. That's why we are saying the transfer RNA has sites for amino acid attachment and an anticodon region for codon recognition that binds to a specific sequence of the messenger RNA chain through hydrogen bonding. As we end, we have the transfer RNA, the ribosomal RNA. The ribosomal RNA are the ones that make the ribosomes, okay? And you refer to them as the catalytic part of the ribosomes. If we look at the eukaryotic organisms, the eukaryotic ribosomes contain 4 different RNA molecules: the 18S, 5.8S, 28S, and 5S are ribosomal RNA. Okay, so the ribosomal RNAs are also synthesized in the nucleolus, okay, and they are transported back through the nuclear pore into the cell, into the cytoplasm. And in the cytoplasm, the ribosomal RNA together with proteins assemble to give us nucleoprotein. So the ribosomes bind messenger RNA and they carry out protein synthesis. Okay. Yeah, we're going to look more about ribosomal RNA as we talk about molecular biology. So we have interesting properties of DNA, and DNA, when it's exposed to heat, can cause denaturation, similar to what we saw in protein. And this time, instead of it being destroyed, we kind of melt. The 2 strands separate because the hydrogen bonds are broken. So it is sometimes referred to as the melting of DNA. This melting causes disruption of the hydrogen bonds, but does not break the phosphodiester bonds. And very interestingly, when the temperature reduces, the DNA strand comes back together. So each DNA has its characteristic melting point, and this melting point depends on which kind of bases are there. 2 DNAs, one having high concentration of adenine and thymine, another one having high concentration of guanine and cytosine. It's no surprise the one with GC will have a higher melting temperature because Remember, between guanine and cytosine, we had 3 bonds in between, while in the other one we had 80, we only had 2 double bonds. So it will require slightly higher temperature to melt the DNA double helix that has predominant concentration of G and C. You can see here this strand. You see these 2 strands. One has a melting temperature of 80 degrees and another one with a melting temperature of 85 degrees. So that is it. I hope you've enjoyed this class. It's a very short summary for nucleic acid structure and function. You can use these questions for revision. Until we meet again, thank you. Bye.